Hello, and thank you for the opportunity to speak here today. Today I'm going to talk a little bit about what we've done looking at urine for biomarkers to track with disease progression in Duchenne Muscular Dystrophy. So Duchenne Muscular Dystrophy is a X-linked recessive disorder which shows muscle wasting throughout life. But before 1990, we see that the average age of death is at around 20 years old. And so recently there's been a use of steroids and improved cardiac management to delay the disease, and we see that the average age of death has been increased to about 25 to almost 30 years. So we see this functionally as functional gains up until the ages of 7 or 8. However, around that time we start seeing functional decline, such as the loss of ambulation in the mid-teens. And at around the later teens, we see functional decline in the upper limbs until death. And so novel clinical endpoints currently used in DMD are all functionally based. Specifically, the 6-minute walk test is used, which is the distance that a boy can walk in 6 minutes. And I bring that up because you see that they're also using this in the time that they have functional decline. And that can be a problem because many of the boys have to leave the clinical trials once they lose ambulation, or they're not allowed in once they lose ambulation. The other thing is that they are boys at the young ages of 5 to 15, so sometimes we just don't get them to comply. And so we see that the endpoints may have some limitations, and we see this with there's no actual cure for DMD at this rate. And so we think that there's still a need for an objective biomarker to track with disease progression and therapeutic efficacy. So our lab focuses on MRIs, which you can see here gives us a nice large field of view, and we can see that it's able to track with disease progression. So I can orient you to the outside of the control is that in the high intense lighter area is the subcutaneous fat, and in the middle is the bone, the femur. We see that that homogeneous gray area is healthy muscle tissue. You can see as compared to the Duchenne that we see, again, subcutaneous fat, but now it's infiltrated with fat and fibrosis into the muscles. I'd like to just point out the muscle that's indicated by the red arrow, that you can see that if you were to take muscle biopsies from one side or the other, you would get a very different story as to how the disease has progressed. And so biopsies not only being invasive can also show that they can't track specifically with the full disease. So the large field of view that MRIs give is very helpful, but also we're able to track disease progression using inherent magnetic properties such as T2, which is the relaxation of the water molecules, where we see that in normal tissue it's much faster, whereas when it's has inflammation or fat infiltration, there's a much longer T2, which I'll talk about a little bit more later. So at a cellular level, the Duchenne muscular dystrophy is caused by a lack of a functional dystrophin protein, and this causes disruption in the membrane. As you can see at the top, that there's a healthy muscle where we see dystrophin binding the intracellular actin at the N-terminus, and at the C-terminus, it binds the dystrophin-associated glycoprotein complex, which then binds the extracellular matrix. And all of this together causes a very stable muscle cell. However, at towards the bottom, you can see that when dystrophin is not present, we see that it's a very fragile muscle cell, and that when the muscle is used in eccentric or muscle contractions, that we see a ripping of the muscle, and that macrophages and repair fibers are brought in due to the influx of other molecules into the cell. We see this ongoing bouts of repair and damage through the boys' lives until the repair or the regenerative capacity is no longer there, and we see that the infiltration of fat and fibrosis renders the muscles unusable. So there's been shown in muscle extractions from mice and humans that there's been multiple different changes in different pathways. However, I'm going to just bring up the two, the creatine phosphate pathway and the TCA cycle. And in the red arrows, you can see that these are the changes that have been seen in muscle tissue as well as serum. And we would like to then look for these changes in a more readily available biofluid such as urine. So our objective was to determine a biomarker for DMD, assessing the disease progression using metabolomics. And I work in a natural history study at the University of Florida, showing that uses MRIs, functional testing, and strength testing to track disease progression in DMD. My part of the study includes collecting urine and serum from these boys, and we've actually consented 90 of them to urine collection so far. I've then separated them into boys that have never taken steroids, those that are currently taking steroids, and controls. And I'd just like to point out that our controls are really low, as it's kind of difficult to find boys that are willing to go through the whole process that aren't in the disease. And so for this point, we didn't really do multivariate analysis. We decided to target our analysis on the pathways that we'd previously seen to then look at the specific metabolites in the urine from the muscle, from the literature that we've seen in the muscle. So I'd like to start looking at the TCA cycle. And so based on previous literature, it's been shown that multiple TCA intermediates have been affected by DMD. But today I'm going to specifically talk about citrate and succinate. So based on the previous literature, we see that citrate will increase in the DMD population as compared to controls, and that succinate will decrease in DMD as compared to controls. But we're also interested to see if either metabolite will actually be affected by steroid treatment. So when we looked at succinate, we saw that there was no significant difference in between control or either of the DMD groups when we looked at our NMR. And so we then did a targeted mass spec analysis, and where we verified that we also saw no change in succinate. We also looked at all of the other TCA intermediates that were found to differ, and we saw that most of them tracked similarly to what's been previously seen in the literature. However, citrate we saw increased in the boys with DMD that were not taking steroids as compared to controls. But those boys that had DMD that were taking steroids were rescued to almost control levels in the NMR. So when we ran our mass spec targeted approach, we then saw that we actually did see those levels actually go back down to control levels. So what this shows is that we were able to see similar metabolic differences that occur at the muscle, but now we can see them in the urine. And we saw that succinate, we did not see a change. However, in citrate, we saw to increase in DMD as it had previously been seen, but now we saw a decrease back to control levels in those steroid-treated boys. So then to focus on the creatine phosphate pathway, we saw that one of the major things that they look at when they suspect that a boy might have DMD is the creatine kinase in the blood, which is potentially up to 100 times more. And so we thought that this could potentially be affected in the muscle tissue itself. So we wanted to look at the metabolites creatine to see if they were affected. And in the literature, creatine is quite an interesting story because it's shown to increase in the blood but decrease in the muscle. And so there's multiple theories on why this could be occurring, but we think that since creatine is a metabolite for healthy muscle, that it would also be able to track with disease progression, and it would be increased in the urine as the muscle tissue was being used. So we did see that there was an increase in creatine in boys with DMD as compared to controls, but we did not see that it was rescued by the use of steroids. So when we looked at an ROC curve to determine DMD versus controls, we saw that the area under the curve was quite high. But again, our ultimate goal was not to see the difference between DMD and control. We wanted to see if it would also track with disease progression. So we in our lab, like I said, we used MRI, and one of the ways we track disease progression is using that T2. So what I'm showing here at 40 milliseconds is a healthy control T2, but the boys with DMD you can see are quite elevated, and we can track that it increases over disease progression, even at different ages. And so we then plotted our creatine concentration as compared to our T2 and saw a general trend that as the T2 increased, we also saw creatine increase as well. But On top of that, we also saw this substructure relating to age. And so as they were younger in age, we saw that they had a more normal T2 and a decreased creatine level. But as they aged, we see an increase in T2 and creatine concentration, showing that we're watching the infiltration and inflammation of the muscle. And we see the creatine being released from the muscle. But after a certain age, particularly around 14 and older, we see that it reverts back, showing that we now can see that the T2 is decreased due to fibrosis entering into the muscle, which has a much shorter T2. And also we see the creatine decrease due to the fact that there's less healthy muscle being able to release the creatine. So looking at creatine as a marker for healthy muscle, and we also wanted to look at muscle breakdown biomarkers, which we've seen 1 in 3 methylhistidines to be in the literature. And so you can see that the 3-methylhistidine is in actin, and through proteolysis is then released into free blood, and through the kidneys is released and excreted in urine. And so we hypothesized that the 1 in 3 methylhistidines will increase in boys with DMD as compared to controls, and that the 1 in 3 methylhistidines would decrease in boys then using steroids. And so in our targeted mass spec approach, we saw that there were large increases in boys with DMD, both using steroids and not, as compared to controls. And so we weren't able to see that rescue again with the steroid use, but we were then looking a little closer at the population that accompanied our boys that were taking steroids. And it was confounded with age, how long they'd been on steroids, and also the disease progression themselves. So we looked at the data that we had acquired and saw that we had a couple of case studies that we had time points both previous to steroid initiation and post-steroid initiation. And you can see in both, we see before the steroid initiation that their 3-methylhistidine concentrations are quite high, but after we see that it is dramatically decreased. And so although it's a very small number, we see that it is potentially a biomarker tracking therapeutic efficacy looking at these 3-methylhistidines. So in summary, the TCA intermediates are shown to differ between DMD and control, and that citrate can differentiate steroid status. We see that creatine may be able to track with disease progression, and that 1 in 3 methylhistidines are increased in DMD as compared to control, and that potentially this 3-methylhistidine is able to be determine therapeutic efficacy. So in the future, we'd like to increase our sample size and analyze more longitudinal samples. We'd like to create a multivariate model using both the global data and MR data to test for disease progression, and analyze serum using both the NMR and targeted mass spec techniques. And so with that, I'd like to thank the Metabolomics Common Fund Supplement to the Parent Grant for the opportunity to do this study. It's given us a lot of ideas and things that we can do in the future. And of course, CSUM and my mentors, and of course, all my lab members. And with that, any questions?
You have time for one question?
I saw you use creatinine as normalization method, right? So creatinine is a major metabolite of creatine. So why you use that if creatine is changing?
Creatinine. That's an excellent question, and it's something that we've actually thought a lot about what we were going to normalize to. So we've actually done it both ways, with and without normalizing to creatinine, and we see similar results. But creatinine does seem to change a little bit between the populations, and we just can't account whether it's a kidney, a problem with the kidneys with the boys taking steroids, or if it has to do with the disease itself. Also, we wanted to be able to compare the results to what's seen in the literature, and everybody seems to normalize to creatinine. So it was just what we've been able to do so far.
Okay. Because I also work with urine, so because athletes, they also like take a lot of creatine. So they suggest, anti-doping agencies suggest to use the specific gravity for normalization. Maybe you can try it, or you already done it.
We have it. We just don't have it for all of them. So it was later on that we started to acquire it. So it's something that we need to look a little bit more specifically at what we have. But we have thought about that as well and have the data to do it. Just haven't done it. Thanks.
Thank you.